Okay, welcome back. So I've labeled this video as Big Data Biodiversity Informatics, but in fact, what I want to do is to begin with the resource that most systematists, insect systematists, use for their work, and begin there and work up from the collections, which is this resource I'm talking about, to the data that they hold and what you can do with those data on the other end, aside from just basic taxonomic systematic research. So let's jump into this. So collections. Insect collections take a huge variety of forms. Sometimes we might think of them in a sort of a trivial manner. Think of a butterfly collection or something like that. But in fact, research collections can be extremely large and dwarf in their magnitude other natural history other types of natural history collections that you might think about. So the important insect collections, aside from, well, let's put it this way. There are some personal collections that are trivial, small, minor, very minor players in the big picture of things. But there are some personal collections that are very large and have been very important over the years. Just to pick out a couple of examples that jumped to mind as I was considering this. The Barry Valentine, who was a former professor here at Ohio State, was one of the world experts on the what was then the beetle family Anthribidae, now I believe called relegated to the rank of a subfamily. But Barry, over the years, built up probably the world's best, most comprehensive collection of anthribids from entirely around the world. Henry Towns was a hymenopterist who from the 1940s, when he began this work, up until he died, I believe sometime in the 1990s, built up this huge collection of Hymenoptera that he eventually incorporated and is now associated with Utah State University. Valentine's collection, by the way, went to the U.S. National Museum of Natural History, the Smithsonian in Washington. And then Henry and Ann Howden were specialists on the beetle family Scarabaeidae, and they also built up this just monstrous private collection of scarabs from around the world. The two of them traveled around the world. They lived in Ottawa, and their collection was so big, and of course they kept at home, and so their house was literally with insect cabinets all around it. When you walked in, it was like walking into an institution almost in some respects. So all of these personal collections, some of these personal collections are very important in terms of the science of systematics and taxonomy and what insect collections are used for. And I'll get into that a little bit in a couple of slides here. Other places that you find the major insect collections are universities, but this is primarily a phenomenon of North America, and even there I think it's primarily a phenomenon of the United States. There's a big collection of insects at the University of Alberta. The University of British Columbia has a modest one. McGill University in Montreal has a modest insect collection. And of course Guelph has a good collection. But in the United States, that's where you find most of the university style collections. It used to be that practically every land grant university where entomology was taught, and that was most of the land grant universities, there was an insect collection associated with that department. These typically started out as tools to assist with identification and teaching, but gradually took on more and more of a role in the science of systematics as their primary function. Couple of universities that are not land grant universities I wanted to mention was the Museum of Comparative Zoology, which I think is kind of a hybrid between a private organization and its association with Harvard University, but that's located in Cambridge, Massachusetts. And one of the other major insect collections I know of at a university is at Lund in Sweden. And that one gains its prominence from the fact that a very important systematist, C.G. Thompson, worked at Lund University in the latter part of the 19th century. And so his collection formed the core for a second century of extensive work in insect systematics and collecting, not only in Sweden, but all around the world. So university collections are facing some difficult times in terms of funding and how priorities change. Universities like to think of themselves as nimble and able to adapt to the changing intellectual climate. And so whereas taxonomy and systematics was a very prominent part of university departments through at least the first half of the 20th century, and well, well beyond that, well up into the 1980s. It has been gradually, and sometimes not so gradually, declining since then. So their relative role is starting to diminish, and some universities are not abandoning their collections, but passing their collections on to other institutions that are prepared to curate them and take care of them for the foreseeable future. Other places where you'll find important insect collections, of course, include governmental agencies, state, provincial, national government sorts of things. So just as a couple of examples, we have the Florida Department of Plant Industries. They have the Florida State Collection of Arthropods, which is located just across the street from the University of Florida in Gainesville. There's the California Department of Food and Agriculture in Sacramento. Those are two non-federal organizations in the United States, and you might be able to infer on the basis of which states these are in that these things exist primarily for the purpose of biosecurity, for quarantine and for protection of the borders from allowing invasives to come in, because those California and Florida, of course, are two major ports of entry into the country. In other countries, similar sorts of organizations whose primary purpose is to protect the economic interest writ large of the country are in Australia, the Commonwealth Scientific and Industrial Research Organization, which some people call CSIRO, or some of the Australians call it CSIRO. You can take your pick. In addition to a lot of the direct technical and what we would call extension capacities that's done in Australia. They house the Australian National Collection of Insects. And then to the north of us is Agriculture Canada, and I pulled out a picture of one of the main building there for the collection of Agriculture Canada. This is the KW Neatby building. This is in Ottawa, obviously at a warm time of year, although there's some of the trees still look a little bare. I've spent many an hour up in one of these third floor windows, probably a little bit closer to this end of the building, working there on what's it called? The Canadian National Collection of Insects, Arachnids, and Nematodes. So again, given its position within Agriculture Canada, its ostensible purpose is to protect the agricultural interests of Canada from foreign invasive pests, and of course in some cases the native pests too, through identification and monitoring and things like that. But in order to do that, they have to put a lot of effort into basic taxonomy and systematics along the way. Now, when I began this slide and telling you I was going to point out where major insect collections are, you probably thought of the things that I'm going to show you next. And that is the private and national museums of natural history that are spread around the world. There's lots of these things, some big, some huge, and some a little bit smaller. Let's take a look at a few of these. Let's begin with the Smithsonian institutions, National Museum of Natural History. So this is located on the Mall in Washington, D.C. And as you'll see with these pictures, a lot of these buildings are used not only for research purposes, but also have a major mission in public displays and public education. So if you've ever been to the Smithsonian, you know inside the rotunda there they have the big stuffed elephant, and then off to the sides, not in the main portions, but you'll find the areas in which the materials that are on display, well, the ones that are on display aren't stored there, but the materials that form the source for the things that go on display are stored in great numbers. And of course they have a major insect collection, which they operate in conjunction with the USDA Systematic Entomology Laboratory. And the Systematic Entomology Laboratory, of course, is one of the main agents for identification and screening of quarantine questions that come in. This is the Natural History Museum in London, right on Cromwell Road in South Kensington. Again, a very ornate, imposing building built in the late 1800s, and it's got a couple of recent additions to it. Most of the space you see here off the front is display areas, and then back in the bowels of the building is where you'll find the collections themselves. The Muséum National d'Histoire Naturelle, the National Museum of Natural History in Paris. Lots of public display areas, collections in the back. Actually, the insect collection is in a building off to the side over here. And the museum itself fronts on the Jardin des Plantes, the garden of the plants. And there's a little zoo off to the edge there too. So it's a very popular area in Paris. What else do we have here? The Zoological Institute in St. Petersburg, situated right on the banks of the Neva River. This is an ornate, sort of not functional light beacon for navigation here. One of my colleagues that I work with, they worked on the same group of insects that I did, worked at the ZIN, they call it, the Zoological Institute. The M comes from the Academy of Sciences, in Russian, of course. So that's another institution with a lot of research going on in it, but most of the floor space is devoted for exhibit space. They have interesting things in this museum, like baby mammoth that was pulled out of the permafrost in Siberia, so they've got it on display. They have Peter the Great's horse stuffed. Now, Peter the Great, I believe, stood about 6'7" or 6'8", and how that huge man could get on what, in his present condition, looks like a pretty flimsy mount is totally beyond me. And of course, so these are national museums of natural history. So these are funded centrally by the government, but they really are not serving, their primary purpose is not to serve a governmental function per se. Just contrasting that with the things like the Florida State Collection of Arthropods and things like that, whose function is identification and extension sort of support services, extension and quarantine support services. Other institutions that house major natural history collections are some private institutions, so things that are not funded by the taxpayer. One example that I've got a picture of here is the Field Museum in Chicago, located right on the shores of Lake Michigan. If you go in there, of course, in their rotunda, what the famous exhibit they have there is Sue, the Tyrannosaurus Rex. But back off the back of the building is where you find the collections themselves. And they've actually got a couple of floors that go underground to increase the amount of storage space that they have. And of course, there are several other, this is the last picture I have of a private institution. There are several others that probably come to mind, such as the American Museum of Natural History in New York City, the Los Angeles County Museum of Natural History, located, of course, in Los Angeles County, and Carnegie Museum of Natural History in Pittsburgh. So there are a number of these in the United States that are found. So insect collections are found in a wide range of places, in private collections, in universities, in state and federal government agencies, national parks. Sometimes national parks do have collections. They're usually pretty modest in size. And then we get into the national museums of natural history, or these huge private museums of natural history. So that's where the collections are held, and that's what forms the resource for people like me to come in and start doing research on the diversity of insects, who they are, how you tell them apart, how are they interrelated. So what are these collections doing? Given such a wide range in their funding sources, in what their goal is, you should not be surprised to find that there's a wide range in their purposes for them being there. Obviously, most of the ones that I've talked about so far have at least a component of their purpose to be to conduct research on the collections, that is taxonomic and systematics research. But I think it's probably fair to say that in many places this is not their primary purpose, except for maybe some of these large private collections that people build up to support their research. All the other ones have other ostensibly public, more public-facing research functions. These may include identification services, just to tell people what things are. Do I need to be afraid of this? Do we need to let this ship offload its cargo of grain since we found these moth caterpillars in there? These kinds of things. They provide identification services. Public displays and education are important. And of course, this is a subject that we could probably spend the whole hour on, about how you effectively do this. Public displays, in order to be effective, can't be static. I mean, certainly some displays kind of take on an aura of tradition. The elephant in the Smithsonian, that's probably going to be there for decades yet to come. But the rest of the museum is constantly changing, and I've seen it constantly change and overall improve over the years with new displays, talking about new things. To keep the audience coming through the years, through their lifetimes, to come and visit the museum. Another important role that natural history collections play is in the support of conservation research. And here I'm talking about the conservation of organisms, so rare and endangered species and things like that. The American Museum of Natural History has a program in conservation science that builds off the expertise that they have in the organisms themselves through their curatorial staff. And then, of course, finally, collections serve as places where these materials, these specimens, can be archived and preserved for the future. I think it's fair to say that, except for large charismatic organisms like redwood trees or giant pandas or birds or things like that, the vast majority of biodiversity on the planet, and certainly the vast majority of the insect and biodiversity, it's what we know about where those species occur, when they're active, what they're doing, what they're feeding on, what kind of habitats they live in. All of these things are documented primarily in the collections held by natural history museums. And so this forms basically the data, the core source data for what we know about all of the insects in the world. Maybe honeybees would be one example where this is not relevant, and perhaps some butterflies, but the vast majority of insects, what we know about them, about where and when they occur and what they're doing, is based upon the data that is associated with the specimens that are stored in these natural history collections. And more than the data, as we've come to find new ways to extract information from the specimens themselves. And of course, I'm thinking particularly of DNA extractions here. Find new ways to get new characteristics, new information from those specimens. And so sometimes even specimens 100 years old or more can have their DNA extracted and sequenced to tell you what that specimen has. We have specimens in our collection. This might be a bit of an apocryphal story, but I had a colleague who was hired in, and this was in the days when DNA sequencing commonly involved using radioactive isotopes as instead of the fluorescent dyes in order to do the sequencing and identify the bases off of a gel. And so he purchased a Geiger counter just to make sure that his lab was clean when the radiation inspectors came around. And just on a whim, we pulled out some specimens that we, for some reason, we have at the Ohio State collection of moths. I believe they were sphingid moths, so they were big. They were quite massive moths, but they were collected in Nagasaki, Japan, which of course, where is where the second atomic bomb was dropped. And he pulled out his Geiger counter and claimed, I think he was just trying to, I think it was more of a good story than actual truth, but we don't need to dwell on that. Let's stick with a good story. He claimed that he could measure radiation in these moth specimens that we had in the collection. So there's lots of information. My point, I guess, is that there's lots of information that can still be derived from these specimens, even after you think you've mined them for all they're worth. But there's lots of data to be gotten, and of course there's lots of data associated with them. In terms of collection sizes, I guess the first thing to tell you is to pay attention to the salt shaker here, because all of these numbers are self-reported and let's say probably a bit self-aggrandizing to emphasize the importance of the collections that they hold, because administrators and others tend to give greater import to world leaders, the biggest in the world, the most extensive in the world, and this kind of thing. So take these numbers with a grain of salt. But the Natural History Museum in London claims to have 30 million specimens of insects in their collection. The National Museum of Natural History in Washington claims to have 35 million. The Paris Museum claims to have 40 million. At the university level, typically people are claiming numbers of specimens in the area of 1 million to about 7 million. If you really held my feet to the fire about the triple horn collection here at OSU, I would say that we have 3 million specimens. But what I'm counting are pinned, labeled specimens in the collection, in the dry part of the collection. So I'm not counting the freezers that we have just stocked jam full of unidentified, unmounted specimens. I mean, there are literally millions and millions in there. I'm not counting those, but I think some of my colleagues may count these sorts of specimens. One story, I don't know the truth of this story either, but it was claimed the Florida State collection was claiming just outrageous numbers of specimens in their collection. And the story was that they were counting unsorted Malaise trap residues. So you put the trap out, take the jar off, and in those days you just put it on a shelf. These days you drop it in a freezer or refrigerator to keep the specimens in better condition. But the story was they were claiming those specimens in their total specimen count, which I guess in a way makes some sense, although it seems like it would be pretty darn hard to actually count the number or come up with a valid estimate of the number of specimens. Or maybe you could have a contest, just like how many jelly beans are in this jar. Tell me how many ants are in this 5-gallon jug of alcohol. This is a picture of the National Museum of Natural History in Washington, just to show how they're trying to manipulate you in this image to show how massive and extensive their collections are. The use of this particular lens in the camera makes it look like that's about a quarter mile away, when actually it's probably about 50 feet down the aisle. Maybe I'm being a little bit too modest there. Then of course they pull out everything and make it look crowded. But it is impressive, the numbers of specimens in this insect collection and any insect collection. You compare the sizes. Plant collections are 1 or 2, perhaps 3 orders of magnitude smaller. Now, granted, the amount of space that a plant sheet occupies is going to be significantly larger, but even the largest collections are dwarfed by the size of these insect collections. So, literally there are millions. I mean, I think just in those numbers, what have I got there? 105 million specimens in just 3 institutions. Even if we allow for some fudge factor in the accuracy of those numbers, if you added up the total number of specimens of insects in collections around the world, it would easily number in the hundreds of millions, if not billions, of specimens. And the key, and we're going to get to this in a second, each of those specimens has information about where it was found, when it was found, who found it. And all of that information is extremely valuable for documenting and understanding the where and when of insect biodiversity around the world. I want to take a short detour here. If we're talking about the collection serving as a place to archive material and specimens as a source of information about organisms, I want to broach the subject of vouchering. This may be a waste of breath, but I think this is very important, and this has been supported by the organizations such as the Entomological Society of America over the years. And what I mean by vouchering is that, I don't think any of you are directly involved in taxonomic research in your graduate career, and probably won't be in your professional career going forward. But you're working on some aspect of the biology of an organism. And unless it's one of those organisms that is so thoroughly known that there's no question, Drosophila melanogaster, Apis mellifera, perhaps the monarch butterfly, and Parapolidina americana. There's a small cohort of insects that nobody has any questions about in terms of their identity. But for the most other organisms, which form the vast majority of them, there's the distinct possibility that as time passes, that when you did your research on species A, as time passes and we learn more, we may discover that species A is actually not one species, but is actually 2, 3, 4 species. I was talking with a colleague in EEOB a couple of weeks ago, and he was one of his big fails in his graduate career is that he had a project all lined up where he was measuring all these parameters of some aquatic insects, and it was going to be the last chapter in his dissertation, only to discover that what he thought was one species was actually, I forget the number he had, but like 6 or 8 different species. And so all the information that he was amassing about intraspecific variation in that organism turned out to fall apart because he was actually dealing with 8 species. Now, he caught it in time. But, once you do a study and publish your results and go on to the next stage in your life, years down the road, it may turn out that what you thought was this species of mosquito actually had 2 different forms that was revealed by, let's say, molecular analysis or behavioral analysis. And what you thought was one species is actually 2 species. Then the question is, did you work with both of those 2 species and didn't recognize the difference? Or did you deal with one or the other of them? And if that's true, which one did you deal with? Which one are your data relevant to? And the way for somebody, the only way really for someone to figure that out is to go back to a sample of the specimens that you worked with and apply the new standards of identification to them. And so it behooves people, unless they're working with one of those, well-known species, to save a small sample of specimens for future researchers to be able to refer to make sure that you got your identification right, or if new species are discovered, to make sure which species you were dealing with in your study. That sample, those specimens, are what I'm calling vouchers. So I think it's good practice, and we always advocate this to students and faculty and department alike, to always deposit a set of specimens from your research in our collection, or it doesn't have to be the OSU collection. It could be any natural history collection, but an institution that's designed to last for years. Encourage you to deposit some specimens with that institution as vouchers. And so basically, the way you do this is first find a collection that accepts vouchers. They all don't do that. Space and curatorial time are always limiting factors in the whole curatorial equation. And so some collections may not have the space to accept vouchers, or they want to focus their acquisitions in a certain direction. But all that takes is a quick email to ascertain the level of potential interest of a collection. And if you, in doing that process, you may have contacted the curator, but if for some reason, they might tell you that they're willing to accept vouchers on their website, contact the curator in charge to find out how to go about this, what the expectations are on the part of the curator, whether your expectations can be met by the collection. And once you come to that agreement, then once you complete your study, send them or give them physically a small sample of the species that you have been working on. If you're working with one species, typically 10 to 20 specimens is plenty. Ideally, you would have representatives of both sexes, just to give an idea. So this is what I thought was Culex pipiens when I did my research. And you may be right, but if you're wrong, then in the future someone can spot that. It's not an error in the sense, it's just that more knowledge has accumulated. But again, then let the rest of the world know that what you did in your research with dealing with this different species. An important thing, though, is that when passing on vouchers, is that I think it's good courtesy, if nothing else, that you, as the donor, pass on specimens that are properly prepared. They're mounted. If it's the kind of species that is slide mounted, then they're on a slide. If it's some kind of species stored in ethanol, it's stored in ethanol. Dried, pinned, pointed, whatever. They're mounted correctly. Second, that they're all labeled, and we'll talk about what goes on the label here in the next slide, I believe. And thirdly, if the paper has been published, to provide a citation to that paper. And so what we do for our collection, and I know some others that do this as well, is we have a special label, colored label. I think it's purple. No, it's green, because I think the purple is for specimens that have been imaged. Put a green label and say that this is a voucher, and then give a tiny little citation of the paper in which it was published, so that somebody coming along later and saying, boy, that was a very interesting result that so-and-so got on this species of beetle. I wonder, did they have the right do they have the same species that I have? Let's go take a look at their specimens. And so you can actually put your hands on those specimens in an efficient and quick way. So that's vouchers. Please consider depositing voucher specimens of your research with a collection. And the Triple Horn collection, of course, is happy to work with you to store vouchers in perpetuity, or as long as universities can think about in perpetuity as we go forward. So basically, what should be on that label? What is the information, ideally, the information that is associated with insect specimens? This is what people in the field call primary species occurrence data. Primary in the idea is that this is the fundamental data on the occurrence of species in the world. So this you can think of this in two components, or maybe three. The first is that there's information about the collecting event. The second, that any additional information is associated. And the third one that just occurred to me is any information about the identification of that specimen. So, collecting event. Here we have a representative label from 1969, which kind of illustrates what the state of the art was in those days. You go back a little bit further in time, and not only the numbers are handwritten, but the entire label is handwritten, and it becomes sometimes it can be very difficult to decipher old handwriting, because you're talking about people writing with crow quill pens. I don't literally mean a crow's feather, but those tiny little pens that you would dip in inkwells and scratch out the whatever you were writing on a tiny little piece of paper that's typically the size of our insect labels. We shoot for something in the area of 1 centimeter by 1.5 centimeters. So if you're depositing vouchers, shoot for that size and not a huge label, because we've got to fit all these into the drawers. If you can't fit all the information on a label of this size, and this is probably 4-point type, then you put multiple labels on there. Please don't print on the back. That's a curatorial nightmare to try to figure out if there's information on the back side of a single label, but people have done it. So basically, the collecting event is the confluence of the place, the time, the agent, and method. And by that I mean the place where the specimen was collected. Oh, just as a side point, the same information is relevant to an observation, and not only a specimen that's going to go in a collection. Now, in the case of a specimen, at least you can go back to that specimen and confirm the identification of it, whereas an observation is, if it's only an observation, you can't do that. I think probably a photograph falls somewhere in the middle between a specimen and a mere observation that's reported. But for all three of those, for a photo, for just a visual sighting, or for an actual vouchered specimen in a collection, we're talking about a place where that organism was seen or collected. When that happened, who did it? That's what I mean by the agent. Who did it? And probably the least important of these four bullet points is the method by which they were collected. So in the case of this specimen, it was collected in Williamsville, Missouri, on the 25th of September 1969. You'll find that there are lots of ways to express dates on a label, and I would recommend that in order to avoid ambiguity in the future, you spell it out as clearly as possible. Because if this was just 25/9/1969, I think everyone in the world would interpret this as the year 1969. And I guess this is not a good example because there's no month 25, but let's say this is 12/9/1969. I can never keep straight of what the order is in terms of month or day, because in the United States we do it one way, and in Europe and many other parts of the world they do it the opposite way. And because I'm dealing with specimens from all over the world, I can't keep straight in my mind which it is. And so I like to do either one of two things. Either express the month as a Roman numeral, which might be a little bit opaque to a user, or to actually write out a 3-letter abbreviation for this, the first 3 letters of the month. Wouldn't take much more space to write SEP here than to have IX period. But the idea is to try to be as explicit as you possibly can, because we do have specimens that, what if it said 4/6? Does that mean that it was collected in April, or does that mean it was collected in June? It could make a big difference in the two, depending on where it's from. If it's from more southern part of its range, then April would be quite a reasonable time to see, let's say, a fly on the wing. Anyway, be explicit as possible here. These are really the two most important elements here. And well, the place falls short a little bit, and I'll get to that in just a second. But for the date, be as specific as possible, be as complete as possible. I don't think, for most purposes, day is fine. I suppose there are some studies where it would be important what time of day, but in all my years, I don't think I've ever seen anybody record that on specimens in an insect collection. The person who did the collecting, from a scientific standpoint, probably not so critical. I suppose from a what's the right adjective? Historical, there we go. A historical perspective, or its perspective on the sociology of science, then that could be important. We undertook an exercise at one point in the old version of our database where we would take a collector and we could follow this person through time, the time being the collecting dates of specimens that it was a man that he collected, and just see him traveling on collecting trips through the world. It was a proof of concept more than actually something that I think had value at the time. Method can be important. I'll tell you how something was collected. Was it collected in a malaise trap? Was it collected in a pan trap, a flight intercept trap? Was it collected on a baited pitfall trap, or any of those kinds of things? Can provide information. I should add that the name of the collector can also be important if it ever arises that there's some question about the data. If the person's still alive, of course you can try to ask them directly, when you said you were in this town, were you actually over here or not? If they've passed on, well, maybe there are records that you can go through to see where they were at a particular time and narrow down the scope of the data that is in question. Now, there's a whole slew of additional information that you could include besides this real core, what I consider the real core here. If you collect specimens that are feeding on a host plant, or are mating on a particular plant, or were reared from an insect host, or any of those kinds of biological factors that you observe at the time of collection, it's very valuable to include that on the label if you can. As a sideline, so what some people will sometimes do will put a field number on the label, which corresponds to a number in a field notebook where the data are transcribed in more detail, perhaps pages and pages of observation of what that specimen was doing before it was collected. Let's see what else. Other accessory information, the type of habitat that you are in. It may be bloody obvious to you when you're doing the collecting that you were in a virgin maple forest in east and western Ohio, but somebody later seeing, such and such a place in Miami County, Ohio, you could just as well have been in a soybean field. So some information on the habitat can be valuable, and any behavior you observe. Lots of these, of course, can be best recorded in something like a field notebook, but then you have to take care that the numbers that you're using on the label can be associated with that field notebook, which means eventually you would want to be archiving that field notebook along with the voucher specimens that you have. So always keep that in consideration. So I wanted to delve in here a little bit more on the place, because where the specimen is collected is, I think, probably the absolutely most critical data element in all of this. So this label from 1969 simply records the place as Williamsville, Missouri. That level of accuracy is probably useful for some purposes, but not others. So the scale of the question that you have in mind is important, and it's important that you be able, if you're going to use these data, that you be able to have some confidence that the data are relevant to the question that you have at hand. So, for example, if you're concerned with issues on a continental scale, then saying that something was collected in Williamsville, Missouri is probably entirely sufficient. On the other hand, if your question deals with microhabitats, is this species associated with this particular plant or something like that, then this is obviously not going to be accurate enough for your purposes. And so the general rule would be to record the data as precisely as possible, and then let people conglomerate data, generalize from your data elements later on, rather than just assuming off the bat that, oh, well, nobody will ever need to know this, or why would they need to know this? You know that you're asking the question, why would somebody ever want to know? Then you are making presuppositions on the utility and limiting the utility of your data. So typically, the place will be recorded in a human readable, or in a word format, corresponding to the political hierarchy. And by that, well, in the United States, that would typically mean that you're talking about state, county, and then some populated place that you're at. And if you can be more accurate than that, you probably should. And there are a number of ways to do this. The most obvious way is to use the geographical coordinates of the area in which you collected. You can get geographical coordinates these days from a number of different sources. GPS receivers, so global positioning system receivers, that you can buy a small handheld GPS, which will tell you your latitude and longitude to a level of accuracy that's measured in a few meters. Or if you don't want to use a handheld GPS, many people's phones or cameras are equipped with GPS receivers that will document where a photograph is taken or where you are. It's always good to try to determine what the level of accuracy of your GPS reading is. Because if it's true GPS, that is using the radio signals from satellites, then the three-dimensional position of the satellites in the sky can impact, or does impact, the accuracy of the measurement. So you might be only accurate to 100 meters, you might be accurate to 1 meter, or you might be, maybe even larger, 300 meters. And so that's going to impact the value of those data. If you don't record the error, on the data, then someone using those data has no idea of whether this is very precise data, because the numbers look very precise, or it should be taken with a little bit of fuzz around the edges. You can also get coordinates from topographic maps. If you can find your position on a topographic map, then simply by interpolation between the coordinates on the sides of the map, you can find out with pretty good precision not only your latitude and longitude, your position, the XY position on the face of the Earth, but also your elevation, which in parts of the world, particularly in mountainous parts, can be critical. GPS receivers are notoriously inaccurate in reporting elevation, so always remember that and try to get your elevational data elsewhere. So let's see, formats. A geographic position can be recorded a number of different ways. The most straightforward is latitude and longitude. And I would urge you to remember that not only is a measure of the accuracy of that measurement important, but also something called the geodetic datum. So when your GPS receiver takes measurements from satellites, basically measuring the distance it is from at least 3 satellites, it is making an assumption about the shape of the Earth, about this oblate spheroid that you're on, making assumptions about the details of that. And there are many different such models, and the way you distinguish them is by a piece of information called the geodetic datum that basically specifies which model of the Earth that you're using. And in some parts of the world, using the wrong geodetic datum, or if someone doesn't report it and you make a bad assumption about which geodetic datum they probably used, can result in a difference in the accuracy, the true accuracy of the location that you have by hundreds of meters, if not hundreds of kilometers in some extreme cases. So it's typically very important. I don't know if your camera is going to report the geodetic datum, but your GPS receiver certainly should. So I think that would be the ideal place for you to get this information. So you can report latitude and longitude either in the format of degrees, minutes, seconds, or degrees, decimal minutes, degrees, minutes, decimal seconds, or decimal degrees. So there are many different ways of recording it. Note that you want to avoid over-specifying. Let's say that you take a measurement and your GPS reports it as in degrees, minutes, seconds, and you want to transform that into digital degrees. That is just, say, 88.75 degrees west latitude. There is an implication in the number of significant digits that you use about the accuracy. So you want to make sure that the number that you are reporting reflects the accuracy that you have. So for example, if it said 88.75 degrees, that implies that you are accurate to within 1/100th of a degree, plus or minus, either way. Whereas if you report it as 88.667, because you're not dealing with base 2 or base 5 or base 10 number, then how many times you take out that repeating digit is an implication about the accuracy, and you don't want to mistakenly assert accuracy where you don't have it. Now in some of the older, both the literature and on specimens, you can find locations expressed in terms of township and range, and this is kind of probably not familiar to most people. Maybe if you live out west, you're familiar with township and range, because that's where it really takes on big importance. But let me explain what that is. So township and range, let's start up here, is basically a geolocation scheme that's based upon a meridian or a line of longitude and a baseline or a line of latitude here. And these shift as you go across the US. So it's important to know what your principal meridian and your baseline are. But then you'll see, for example, in this string here, we'll get to the beginning part initially, but this part here refers to township T and range R, and we can translate that here because we see that in this quadrant defined here, this is a township, the square, that it's divided into 16 squares. And here we have, if we're looking for township 2 north and range 1 east, that means with township 2 north and range 1 east, we're talking about this section of that entire township. And so that specifies that. Now that square there is actually, well, depending upon the accuracy that you desire, that may be good enough. But if you want more accuracy, this is 1 square mile in area. So this here is showing the breaking that one section down into 36, I believe, yes, 36 separate subdivisions, numbered consistently this way, kind of interleaving back and forth here, 36. And one of these sections, so township, range, and then this is a section. These are sections within the township and range. Can be looked at this way. One section is an area of 640 acres. And typically people refer to these by their position within that. So in my example that I pulled up here, the northwest 1/4 of the northeast 1/4 of section 8. So if this is section 8, the northwest 1/4 of the northeast 1/4, so we're going most specific to least specific. So northeast 1/4 would be this area here, and the northwest 1/4 of that would be this area right here. So to step backwards, then in this picture of the township, the northwest 1/4 of the northeast 1/4 of section 8, township 2 north, range 1 east would be right about here. Whoops. On this, if this is section 8, and of course we're going to have many sections throughout the country. So this is a way of specifying areas. You typically find them in land grants and things like that, in plats, in land records that they use. But you can find these very commonly in insect collections. So that's how you interpret what the township and range designation means. And then you can translate that into latitude and longitude. So, biodiversity informatics. Basically, I guess the first thing to note is the last point, or the middle point I've made on there, is that I'm not talking about bioinformatics. I can't tell you how many conversations I've had with people who immediately equate these two things because they're familiar with bioinformatics through medical applications, molecular biological applications. And typically bioinformatics is dealing with the analysis of the information found in biomolecules, so sequences and things like that. It's a wide field, obviously, and that's not the entire definition of it. But in terms of biodiversity informatics, the primary data elements I'm talking about are those primary species occurrence data, the time and place, the method, and who did it that's involved. The identification of the organisms, so both the taxonomy and their classification are elements of biodiversity informatics. And media can be data elements in there too. And so basically it's using informatics techniques to go from data to actually understanding what's going on, to use these techniques, agglomeration of data, pulling it apart, analyzing it. There's a whole range of techniques that you can use. And I got into this because I was interested in geographic information systems, and discovered early on that although there is a fair amount of data, at least in those days, was a fair amount of data available, things like plants and some, birds and mammals and things like that. For insects, there's actually very little data available in order to do analyses, to ask questions of them. And so in insect collections, it becomes a big issue about how do you acquire these data in a cost-effective manner. Remember, we're talking hundreds of millions, if not billions, of specimens. And so even if each individual data point that you capture is a penny, pretty soon you're talking huge amounts of money. How do you store that information? How do you manipulate that information? And then how do you share that information with the rest of the world? This is all subsumed under the field of biodiversity informatics. Now, typically the data are captured locally, and owned locally, so that people have control of their data. But if I went through and captured all of the information in the Triple Horn collection, that would be very valuable information for me for management of the collection and for other local purposes. But in terms of answering a scientific question, I probably don't have a good complete set of data in this single collection. I would need to amalgamate information from other collections. And so conceptually one could do this through an internet interface that queried multiple collections at the same time. But a more efficient way would be to aggregate data on a regular fashion, to say, I'm going to store all the data on such and such, I'm going to store all the data that we have on beetles. So I'm going to go out at regular intervals to all of the collections that maintain internet accessible databases, gather a copy of their information, store it locally, saving the provenance so people know where the data came from and it can be used. Spam phone call. But if somebody wants to get at the data, then they only have to go to one place. And so people who do this, or organizations that do this, are known as data aggregators. And there are several of them around the world, some with very specific focus, such as FishNet. It's a place where you would go to get information about fishes. There's a VertNet for vertebrates and things like this. But two of the more general organizations that I want to mention, the first is the Global Biodiversity Information Facility, or what is lovingly known as GBIF. GBIF is an international effort that started from the Organization for Economic Cooperation and Development, which is basically an organization between North America and Europe. To the people promoting this was able to convince the OECD to set up GBIF, and it's been in operation now, I think, for about 20 years or so. It's headquartered in Copenhagen, and countries have membership in GBIF, and it has a scientific committee, it's got an outreach committee, and things like that for its purposes. But its primary purpose is to aggregate data from collaborators all around the world into a single place, and to provide access to those data in real time for people to use. Within the U.S., the parallel organization is something called iDig Bio. The dig is digital and bio is biology. I don't remember what the I was. Maybe it was just an homage to Apple products at the time. I don't remember. So actually, these organizations, you might think that one would feed to the other. That is, GBIF would get its information from iDigBio, and iDigBio would get its information from individual data stores. In fact, they kind of work a bit independently in that, for example, our collection provides information directly to GBIF and directly to iDigBio. So there's still lots of things that need to be ironed out in terms of data flow and data ownership and these kinds of things. Let's take a look at the interfaces for these two aggregators. So this is the GBIF homepage. One thing I wanted to point out on here, occurrence records. The total number of occurrence records that GBIF has cached. That is, this is not the primary place where the data are stored. They are stored by the institutions and individuals that are sharing their data. So they have ownership, and they allow GBIF to aggregate the data and put them together with others. But the point here is that the number of occurrence records currently is 1,658,572,354. Let's try that again. 1,658,572,354 records. Truth in advertising. I didn't do the math to be able to say the majority of data, but a huge chunk of those occurrence records are observations coming from projects like eBird, where people make observations, for example, the feeder surveys of which birds are coming to their feeders. They record this, send it to eBird. eBird transfers it on to GBIF. iNaturalist with observation records there. So a significant component of those 1.6 billion records are observations, which means that if an identification was made incorrectly, if there's an image, you might be able to rectify that. But if there's no image, then you have no way of knowing if that identification was correct or not. You can only come to that conclusion by comparing that data point with the mass of, hopefully the mass of other data points to see if it's consistent or if it's a significant outlier. With that caveat in mind, we see there's 56,748 data sets coming from 1,600 different institutions around the world. And this is I don't know if that's a big number or not, but this is the number of peer-reviewed papers that have been published that cited GBIF as at least a partial source of the data. And 5,500 papers. That's a good chunk of literature, and hopefully that will continue to increase. Hopefully the rate of increase will also increase through time. So we can go in and in the search box here, type in well, you could type in a place, you could type in the name of a species. What I did for this demo is just to type in a name of a species. And I put in Polistes dominula, I believe. Let's see. Oh, I was going to introduce iDigBio. I guess I got mixed up on my order. We'll get to the Polistes dominula example in a second. So this is the American counterpart. For all you Latin American citizens, this is the United States counterpart to GBIF called iDigBio. Oh, integrated digitized biocollections. Boy, I didn't get that right at all, did I? In the same way, though, you can go in and search their data portal here to find out what records that they have recorded. And you can see they're also doing lots of other stuff to provide means of using these data besides the minute, boring, very specialized, arcane scientific studies. Education. How can we use information from the specimens in all these biocollections, not only the primary species occurrence data, but images and things like that, for education, for public outreach, for entertainment in some ways. Why not? That could be just as valuable an effort. And also a lot of effort to facilitate all the collections within the United States, and there's hundreds and hundreds of them, to store their data and be able to share it with iDigBio and GBIF. Okay, so here's my Polistes dominula example. If you're not familiar with it, Polistes dominula is an invasive species of paper wasp. It's a European species of paper wasp that has been introduced into the United States, I believe, I forget the exact timing of it, maybe the late 1980s, 1990s, something like that. In GBIF, here is a graphical representation of all of the specimens of Polistes dominula that they have cached records for. By cache, I simply mean that they store a copy of it. And so you can, I'm not going to do this, but you can zoom in on this to get a better idea. So this is the native distribution. Here's how it's been introduced in the eastern United States. Looks like it was introduced perhaps a second time in the western United States. It was introduced into Chile, which is largely temperate in climate. Not sure what I think about this record in Cape Town. It's into New Zealand, and I'm blocking the records that are in Australia. So you get an idea of the geographic distribution of this creature. Now, this gives a human viewer a visual idea of its distribution. You can also download the data directly. So there's a lot of different formats you can get this. This is just a display, but you can download those as CSV files or XML files or whatever you want in order to go in and manipulate them later on. So 25,407 records of Polistes dominula from around the world. And that most of the major insect collections are at best have a small fraction of their total specimen holdings digitized. So this number will increase dramatically as efforts at digitization, that is capturing the data in digital form, continues. This is an iDigBio interface doing the same thing, except this time I searched for the southern pine beetle, Dendroctonus frontalis, showing its distribution here. And I'm not going to zoom in or show you the details on this one. They have generally fewer records here. They thought it was interesting. We want to concentrate on the green records here. These are ones that they think are going to be relevant to the search that you did. Tarsonemus cranci, I believe that's a mite. Dendrolelaps is definitely a mite. I don't know what the other two guys are. These look like biological associates of the southern pine beetle. So once you get these data, of course, you have to assess them for their what's called fitness for use. Are they identified accurately enough for you? For the question that you have in mind, is the geographical location specific enough? Highly resolved enough. And of course, then you have to also eliminate bogus records. Not sure what's going on here in Tanzania. I'm pretty sure I know what's going on here because that, if I'm not mistaken, the geographic coordinates of that specimen are 0, 0, which probably means that it has no latitude and longitude on it, but whoever shared the data, instead of having a null value for latitude and longitude, had 0, 0, which of course is really a place, 0 degrees latitude, 0 degrees longitude. So on the prime meridian and on the equator, bogus piece of data. You must always analyze the quality of your data. So what can you do with data like these? Well, one application I want to just spend a couple of minutes, probably 5 minutes or so here, talking about is ecological niche modeling. The idea here is that we can use the occurrence data of specimens in order to make an inference, or is it a deduction, inference about the fundamental niche of that species. And just recall from your basic ecology, a fundamental niche is defined as the full range of environmental conditions, environment encompassing both biotic and abiotic, that a viable population of species can occupy and use. And the idea is that if I have these records of where a species occurs, then I can look at those spots on the face of the earth and determine what the values are of a whole range of environmental variables. What's the annual precipitation? What is the annual temperature range? What is the soil type? Just go on and on and on. There's practically an infinite number of environmental variables. So if I can then get the environmental variables of interest at the spots where this species is known, or at least we believe it occurs on the basis of the specimens, in specimens or observations in data aggregators like GBIF or iDigBio, then that will be an estimate of this species fundamental niche. So I've got an envelope, an environmental envelope with the values of all these variables. I can then go and look on the entire face of the Earth, or whatever the area that I'm interested in, and say, where are places that fall within this same environmental envelope? And those places would be areas that are suitable for the species to occur. And that is called an ecological niche model. And here's just a graphic representation of the idea. So we have species occurrence records up here, then a series of environmental variables expressed. We're expressing them as maps, right? So this is sea surface temperature, nitrate concentration in the water, so salinity. So this example was dealing with some sort of oceanic creature. It looks to be hugging the shore along here. And then basically plot those in x-dimensional space for how many variables I have here, and then look and find what other places besides the ones where it was collected, which other spaces fit within that same envelope. And those would be places that should be suitable for that species to occur. So this has become a very popular and rapidly becoming an ever more complicated exercise. So a number of questions occur with niche modeling. I mean, the underlying logic is sound, but there's a lot of areas in which we need to be careful. For example, I said we're going to measure the values of environmental variables at the sites where the specimens were collected. Which variables? There is no there's no way I can tell you which ones are the important variables. One of the early modeling algorithms was something called BioClimb. You can still use BioClimb. There's an R library for BioClimb, which basically used data only from precipitation and temperature. And it was originally designed to look at snake distributions in Australia, but it's been widely used all over the world. And then, take combinations of precipitation. We'll take various values of precipitation, maximum precipitation, average precipitation, the precipitation associated with temperature, and the same thing for temperatures. They had like 18 combinations of either precipitation and temperature individually or in combination with one another, and then use those to figure out what the climatic envelope would be within which those areas would be suitable for the species to occur. But, are those actually relevant to the species you have at hand? There's no way to know that a priori. One alternative, also a very early algorithm, used what's called a genetic algorithm, which is a form of artificial intelligence, to basically take a subset of your data and construct a model using a series of variable, and then compare that to the rest of the data, see how well your model corresponds to the rest of the data that you observe, and then change your model and go back and see if you got a better algorithm. So this was more robust in the sense that you could use anything. It could be temperature and precipitation, of course, but it could also be human population density or the level of nitrates, and this could be anything in the world. And so basically you're trying to find which environmental variables best predict the specimens that you, the records for the specimens that you have. Another issue to be concerned about concerns where specimens were collected. If you map out specimens, they seem to have a very high correlation with the existence of roads. So access to areas will also predict where the specimens are coming from. So that would be a variable. But conversely, the roads provide access so you can get material there. Does the absence of a record have any meaning? And I think in the case of most specimens from natural history collections, absence of data does not have significance. You can imagine, of course, some highly detailed surveys that take place where you can be certain, within certain confidence limits, that something is not there. So in some cases, absences are meaningful, and others they are not. Ideally, after you construct a model, no matter which algorithm you use, and as I point out in that last bullet point, there are a ton of different algorithms, and now there are tons of R libraries that make it pretty easy for you to go directly from GBIF data to producing a model of species distribution, or the ecological niche. But after the prediction, ideally you would go back to the areas that you don't have specimens from, but that are predicted to be suitable, and look and see if the species actually occurs in those areas. That would be the gold standard for the accuracy. So let's see. Let's go to the next slide here. Try. So people use these ecological niche models for a variety of purposes. I've seen papers where they're looking at rare species that might be found in just might be thought to exist in just a small pocket of area, and you want to know, well, where are there other areas where the same species might exist? The example I'm thinking was some species of parrot in Mexico. So you could make predictions about where where rare species might occur, or B, where you might introduce a rare species in an attempt to reestablish it. The example on this map is another common use, and that is to look at the potential of invasive species, the potential geographic spread of an invasive species if it overcomes the distributional barriers that it has. So in this example, this is Cactoblastis moths. So these are moths whose caterpillars feed on prickly pear cactus. And Cactoblastis is a species found in southern South America. And these green dots here show you where the specimens originated from. So what is that? Northern Argentina. It's probably Paraguay. Some specimens here are at least up to the border there. So if you look at the environmental variables in these sites where the specimens originated from, and you ask yourself the question, well, what if specimens from this area got out and somehow were introduced into North America? How far would they spread? So they took the values of these environmental variables and asked, where in the rest of the Americas are areas that also fall within the max-min or 95% confidence, or not confidence, but the 95% limits. Where do 95% of these points occur, the range of that environmental variable? I'm sure that sounded confusing. Just think of it as the maximum min. What other areas fall within those limits for those environmental variables? And typically the color coding indicates a higher or lower level of suitability, and red in this case being a high level. So they're extrapolating from the environmental conditions in which these specimens occur here to say, where could they exist? And in North America, you can see they can spread quite widely through the Florida Peninsula, up into all of the prickly pear that gets up into Georgia and maybe the Carolinas, Texas, over into Mexico, Arizona and New Mexico here. So you can see the potential range expansion of this moth if it were accidentally introduced into the US. I'm sure you can think of other parallel sorts of uses of ecological niche modeling. If you're looking for a biological control agent, where should you go look? Well, it depends on where you want it to be introduced. And so find areas that match in the environmental variables and get your biological control agent there in order to bring it to the US or wherever you're introducing it. So they're very popular and a lot of utility, or a lot of theoretical utility, but it's really easy to get down into the weeds about lots of details to be concerned about in order for these things to be accurate and useful. We could spend, maybe it would be a great idea to have a like a 1-credit seminar course talking about ecological niche modeling and all the ins and outs and how to use it and concerns to be about it, because they're becoming very, very popular. And now there's a billion records to play with in order to ask questions about the geographic distribution and the biology of organisms around the world. And of course, you can have fun with it too. So here we have on the left the distribution of purported observations or the observation of footprints of Sasquatch in California, Oregon, and Washington, state of Washington, from which we can derive a projected distribution of the species of Sasquatch in the western United States. So as it turns out, that distribution matches pretty closely. This was actually published, by the way. You can see the citation down there in the Journal of Biogeography. It was the authors then compared this projected distribution of is this a hominid? I don't know what family Sasquatch would be classified as, but the mountain man, with the distribution of black bears in the western United States. And it turns out it's practically identical. And the same variables, this was using an algorithm called Maxent, and the same environmental variables that are important in determining this distribution, where the darker colors are the more likely areas. Looks like he likes San Francisco. This matches pretty much the distribution of western black bear. And so their conclusion was that probably most of the so-called sightings, or the footprint sightings of Sasquatch, were probably black bears that somebody saw in the woods. I thought that was a fun paper. So, quick summary. I believe it's extremely important to recognize that the specimens and natural history collections collectively document the distribution, time, and space of most of the organisms of the planet, most of the things that we share the Earth with. What we know about where and when they occur comes from specimens and natural history collections. So these collections are important to us. Primary species occurrence data it's a combination of where something was collected or observed, when it was collected or observed, and then some additional data like who did it, how they did it, and then you can add other data elements to go along with it. But time and place are the two most important ones. Another plea, vouchering is a critical but often overlooked step in the research process. The vast majority of entomologists do not voucher their specimens. They all tug their forelock and say, oh, I really should, but I just didn't get around to it. Plan ahead in your research. Plan to deposit vouchers. Protect the integrity of your hard-won results for the future. There are national and international efforts to amalgamate and provide access to this massive data and exponentially growing massive data that we have on primary species occurrences. And the big boys here are GBIF globally in the United States, iDigBio. The Atlas of Living Australia is a big effort in Australia, and other countries have efforts, and regions have efforts going on as well. These are basically open access. You can get access to these data with some of them ask you to some basic ethical considerations about how the data are to be used, but you can get to these billions of records to ask and hopefully answer your own question. And then finally, ecological niche modeling is one way to be using this data that allows you to address very interesting questions about where species occur, what determines their distributions, what the potential ill effects of an accidental introduction could be, conservation questions, and the like. It's a rapidly growing area with methods. I haven't talked about specific methods there because it's just changing at a lightning speed. There's a ton of literature there, very interesting literature, and very, I think, productive and valuable uses of our primary species occurrence data. So specimens and collections are worth more than just what I, as a taxonomist or systematist, might make out of them. There's lots of fundamental biological questions and economically important and medically important questions that can be asked using the specimens in these collections. So don't underestimate their value. So with that, I'm going to sign off and I wish you well, and we'll see you again soon.